Welcome to State of the Science number 3. This is a new way of talking about research in the field of type 1 diabetes. We have a global audience of scientists here today, and we thank everyone for joining. We'll first talk with 3 scientists who anchored the discussion and set the stage for the conversation, and then we'll interact with the audience. Then 3 panelists will join to extend this discussion, and then again, we engage with the audience. In this way, we kind of have a town hall flavor of the discussion. We'd like to take a moment to recognize our fantastic sponsors, 10x Genomics, Prevention Bio, Imsice, Vertex, and Inspiro. These are based both in the US and in Europe. We really appreciate their support. These organizations are at the forefront of cutting edge science, and without their support, we really couldn't even bring you the State of the Science. So you can find more about these innovative companies on our website or at our Ask the Expert series, both live and archived on our YouTube channel. So just a few pro tips for attendees. If you would like to submit a question, place it in the Q&A, and you can submit the questions throughout any questions throughout the presentations. Participants can upvote favorite questions at the end of the session. We will answer more questions as time allows. This event is being recorded live on the Sugar Science YouTube channel and housed thereafter for future viewing. So our first set of speakers, Marian Rewers is an MD PhD at Colorado University of Colorado, and as such, he has a unique perspective that intersects with both the clinic and the research setting surrounding type 1 diabetes. Stephen Rich at UVA, University of Virginia, his research focuses on the genetic basis of type 1 diabetes with establishment of the T1d or type 1 diabetes genetic consortium, in which over 40 loci have been mapped and T1d causal genes have been identified. And also Jill Norris, PhD, MPH, also at University of Colorado. Her research focuses on the influence of the environment in the development of autoimmune diseases, including type 1 diabetes, celiac disease, rheumatoid arthritis, lupus, and multiple sclerosis in genetically susceptible individuals. So welcome everyone. And for the first set of speakers, the second set of speakers, I guess I will introduce also now. And then I'll just highlight their names as they join us in the second section. Heikki Hyoty, MD PhD from Tampere University. His principal research is the role of microbes in immune-mediated diseases, particularly type 1 diabetes. Kyle Gaulton, PhD from UC San Diego. He's bringing his background in computer science, genetics, and molecular biology to bear in chromatin mapping that's involved in T1 risks. Luping Zhao, PhD, MS. He's a biostatistician with a background experience in bioinformatics, epidemiology, and genetics, and he aims to accelerate the translation of big data technologies and findings to the clinical setting. And finally, Douglas Melton at Harvard Stem Cell Institute. He has a goal to cure type 1 diabetes using islet cell implants. And Mark Atkinson here will join us from the University of Florida. His research is broad in scope, but it's directed really at identifying a prevention or cure for type 1 diabetes. So welcome all of you. Thank you for taking the time from your busy schedules to join us. A couple of questions for the panelists to consider in the course of our discussion include, what are the main conclusions you draw from the genetic analysis of type 1 diabetes? Secondly, is there anything else more to be learned from large-scale genetic analysis of T1D? And then finally, and we can revisit these throughout the conversation, can the genetic findings be used to prevent or treat the disease? So I'm just gonna, start with Dr. Rue. Would you like to set the stage with your impression of where things stand in terms of what is cutting edge in your sort of T1D research wheelhouse right now?
Thank you, Monica, for your kind introductions. I think we are living in very exciting times in terms of type 1 research and even from the perspective of the patients. So I would like to just mention 3 main areas of research and translation activities that we are engaged in and we may like to talk about today. Number 1, there is a continuing effort to improve the lives of people who already have type 1 diabetes. And there are very impressive accomplishments in this field, including ability to apply continuous glucose monitoring to any patient at any age, at any time of day, overnight, leading to the development of artificial pancreas and parallel efforts to develop newer, better insulins, as well as adjunctive treatments, for example, SGLT2 inhibitors are already on the market. So all of those lines of research and implementation are meant to lower the daily burden of life with diabetes for our patients. However, this is not enough. We would like to prevent diabetes, and there are several areas for prevention. One is very active. The pre-symptomatic diabetes is now staged into stage 1, which does autoantibodies, but normal blood sugars, and stage 2, with already some abnormalities of glucose tolerance. And there are a number of really exciting candidate immunomodulators that have been tried or are under investigation to slow the process of progression from islet autoimmunity to clinical diabetes, or even to abort this process altogether. There are also some new potential trials of primary prevention on the horizon. One with Coxsackie virus vaccine that has been talked about for a long time and now became a reality, may become a reality for kids next year. So in the area of prevention, a lot of action, a lot of exciting news, and genetics fits here perfectly because one treatment, one prevention will not fit everybody. So there's a huge need for precision medicine and precision prevention down the line. And finally, it's a cure. We really need to not only help people who have diabetes and prevent new cases, but really get rid of type 1 diabetes as much as possible. So beta cell replacement therapies based on stem cell research and beta cells derived from stem cells are at the most promising direction here. But it has to be coupled with harnessing continuing autoimmunity in patients who already have diabetes.
Yeah, that was an excellent summary of, I think, where where we are, where we need to go. So thank you. I'd like to sort of like also invite Dr. Rich. Would you like to weigh in on what's new in your realm of T1D research and comment on what you think is the state of the art right now, currently?
Sure, thank you, Monica. Well, again, I'm focused more on the genomics side of things, and I think it's important to remember that the risk of type 1 diabetes is about equal genetic versus non-genetic, which many cases means environmental. So, I'll let Jill address the environmental component. In our genetic aspects, we've really extended what we think we know about the genetic basis of type 1 diabetes now over anywhere between 75 and 100 loci, and worked in fine mapping that. I think the real critical areas now are related to the precision medicine aspects of prevention, prediction, prognosis, and therapeutics, and understanding the genetic basis underlying each of those. In addition, I think there's been a great interest in sharing of information and collaboration, and Kyle can actually talk to that as well. But one of the key things is we've got these variants that seem to be associated with risk, and now what's their function? And that goes from variant to function. What are the key targets of those variants, whether it's a gene next to it or a gene farther away? What tissues, what sort of specific features of those variants are important? And it gets beyond genetics into transcriptomics and epigenomics, and using single cell as well as bulk approaches, and thinking about the initiation, which genetics may have a different sort of characteristic in terms of initiation of autoimmunity versus progression versus the outcome, and what sort of perturbations, either local or global, that actually happen to address risk. So those are some of the areas, and it's really important to think even back in history of type 1 diabetes, we're thinking about viruses as being a triggering mechanism. And Heikki can certainly address that much more robustly than anyone. And so thinking about that gene and environmental trigger interaction is going to be critical, I think.
Yeah, that was a great summary. Thank you very much. And then I'm going to ask, I would also like to ask, Jill Norris, Dr. Norris, what's happening in your area? How can you weigh in on, what you're doing intersects with what we've been talking about, how genetics and triggers interplay with type 1 diabetes?
Thank you, Monica. Sure, it's exciting times in terms of examining the environmental factors in type 1 diabetes, because we have these prospective cohorts such as Teddy, Daisy, Dip, Baby Diab, and Suya that have provided the prospective samples and information that is required to look at the environmental factors in type 1 diabetes. They also provide the samples for us to look at sort of more big data omics kind of questions, like the microbiome and the metabolome and proteome and the genome that Dr. Rich mentioned, and the epigenome and transcriptome. All of these, I think, are necessary to work together to really understand the interplay of the genetic susceptibility and the impact of the environment on the individual. We probably, all of you know that the incidence of type 1 diabetes is increasing over time, and it has been for quite a while. And that's one of the main pieces of evidence that suggests that there is an environmental factor. And of course, it's overlaying a genetic susceptibility. But if we want to manipulate things, perhaps to prevent or delay the disease, we need to explore what environmental factors are important. So what have we learned? Dr. Rich mentioned sort of the importance of gene-environment interaction, and there are a couple of strong examples of that from the Teddy study. One observation was that kids who are taking probiotics in infancy had a lower risk of islet autoimmunity, the preclinical disease, but it was particularly strong in those with a high HLA risk genotype. Again, you didn't really see it in the one group, but you saw it in the HLA high risk group. Another example is vitamin D levels. We found in Teddy again that lower vitamin D levels is associated with increased risk of islet autoimmunity. But really this was seen primarily in those who had a particular variant in the vitamin D receptor. So it suggests that it's more complicated than simply looking at environment alone. You really do need to look at either genetic susceptibility or some other genetic enablers, if you will, of the environment. Similarly, we've seen associations with vitamin C or circulating ascorbic acid and omega-3 fatty acids, again, suggesting perhaps inflammation might be important in the etiology of the disease. And in fact, just recently, there was a paper published showing that oxylipins, which are markers of the body's ability to resolve inflammation, are associated with the appearance of type 1 diabetes. So again, using the information that we can collect in these larger studies, prospective studies, to interpret how environment may be working and how the body is able to either fight it off or not, is going to be key to us understanding the etiology of the disease.
Yeah, well, you really framed a very complex sort of analysis there beautifully. So thank you. I mean, it really, I think it's going to come down to this understanding, like you said, of this very big picture. Okay, I'm going to actually just do a quick poll to our audience, and the question is, how can real-world data best add value to the role of genetic susceptibility and triggers in T1D? So I'm just going to launch this. I'm going to see what people say. It's anonymous, and the panelists are invited as well to vote. So let's see if anyone will vote. Attendees are viewing the question. So the answers we've put out there are, sort of CGM data, Jdrf, biomarker testing initiative, blood repository from all newly diagnosed T1D patients, geolocations of newly diagnosed patients, microbiome repository of newly diagnosed patients, or other. So this looks like people are, the Jdrf biomarker testing initiative is out in front. That's very, very interesting. I hope more people will vote. Okay, here we go. We're taking their time. And then we have a couple of questions from the audience. Again, trying to make this as much of a sort of a town hall interactive experience as possible. So yeah, it looks like, I'll stop at 1 minute. It really looks like the Jdrf biomarker testing initiative is in front at 61% in answering the question, how can real world data best add value to the role of genetic susceptibility and triggers in T1d? And now it's even getting higher, 63%. And then I say the second one looks like blood repository from all newly diagnosed, which I think is sort of evolving, is happening and evolving. So that's great. Let's talk a little bit about that. If we'd like some of the anchors to sort of weigh in on that, or what their thoughts are about this poll.
Maybe I will take the first stab on this question. So we picked, majority of us picked Jdrf biomarker testing initiative, but actually, and I did too, but actually I know quite little about what it means today. Jdrf has been at the front of the action to support biomarker studies, but it's hard to tell what it means this year and next year for the community. I think Jdrf alone will not be able to really foot the bill for the studies that need to be done. And such studies actually are already ongoing and collecting a lot of samples that can be utilized for biomarker studies. Let me just mention a few: TrialNet, TEDdy, the cohort studies in Europe like DIP and DIPPES and Baby Diab, and in the US, our DAISY and Do It in Seattle. So those studies can contribute to thousands of samples that are underutilized. What's needed really is an honest broker, somebody who will be well-funded and organize the repository and the underlying database and facilitate the sharing of the samples and metadata.
Yeah, that's well said. I think that, like Mark Atkinson did with the formation of NPOD, as a big tissue repository, perhaps another one needs to be created to kind of circle the wagons on all these different data sets in repository. So great. I'm just going to share a question that was answered. And that question was, as for the variants, like a few in T2d, do the panel know of ongoing work indicating gene mutations and variants protective in T1d? And the question was answered by Stephen Rich. If you want to just read your answer and tell, kind of elaborate, you're welcome to do that.
Yeah, I don't know what I wrote, so that's, I'll start over again. Basically, I know that there are people, including Aaron Michaels in Denver, who are actually, we're working together to look at those individuals with type 1 diabetes, but with protective HLA risk, the VQ0602s that really shouldn't get type 1 diabetes, but do. And so if we can understand sort of what the modifiers are of that overcome protection, or even understand that protective mechanism a bit better, we could maybe design better interventions or even therapeutics. The other genes that are associated with type 1 diabetes, because HLA has half the genetic risk, it has a huge impact. The other genes have much lower effect sizes. So even though they may be protective, like some variants in TIC2 or PTPN22 and others, if you can figure it out, that's helpful, but it doesn't have the same impact as HLA.
Great. Well, thanks for answering that question, and thanks for asking to Vishal. I'm gonna switch gears for a minute. We're gonna welcome the sort of the second wave here. So join me in welcoming the following scientists that we introduced earlier: Dr. Heikki Hyoty at Tampere University, Gaulton at UCSD, and Zhao at the Hutchinson. So as panelists, I wondered if you can weigh in, where you see the state of T1D science regarding this role of genetic susceptibility and triggers in T1D right now. And so I'm going to ask Kyle to answer first.
Sure. So I think, as Steve said before, one of the key things about type 1 diabetes genetics that we've learned is that there's major risks from HLA, but then of course many additional risk loci that have been discovered. Some have modest effects, but most have small effects. But they have value in terms of understanding the biology of the disease. And I think there's an increasing appreciation for the variety of mechanisms through which these variants likely affect disease risk immune cells. But clearly there's a role for intrinsic beta cell function, increasing evidence that the exocrine pancreas plays a role, perhaps causally in the development of the disease. And so I think it's a real opportunity to understand through these variants the biological mechanisms of disease. I think it's also a big challenge given that most of these loci are not mapping directly in the gene, and so therefore not so obvious in many cases in terms of the genes and their downstream mechanisms. So I think it's a real challenge to link these variants to the genes that are being affected, but it's also an opportunity because perhaps we can learn more about the cell types that are affected, the systems, the regulatory networks that are affected. But I think it's a real challenge and requires detailed genomic studies, as Steve said, to link these variants to the genes that are affecting. But there's a ton of biology to uncover. And yeah, and I think particularly in the case of the beta cell, I think it's become clear that there's some interaction between the risk factors that affect beta cell function and the local environment around the beta cells. So exposure to cytokines, other stressors that affect beta cell function during progression to the disease. And risk variants, seem in many cases to interact with those changes during disease progression. And so I think understanding those processes, understanding models that effectively capture that, I think will be important, specifically in beta cells, to understand how risk is mediated. But I think it can tell us a lot about biology and then likely therapeutics that could be targeted to manipulate these pathways of genes.
Yeah, that's great summation. And let's have, we're gonna have the three of you weigh in and we'll discuss with the total group. But Heikki, could you, Dr., let's see, I'm sorry. Heikki, could you please weigh in? Dr. Hyoty, I'm sorry if I missed.
Yes, yes, thank you. Thank you. It's a difficult Scandinavian name. So I think, of course, I agree very much with the previous speakers and about the genes and also the function of the genes, which is less well known. And I think that's one knowledge gap. We need to go to that. And I think one of the ways how we can get that information is to study the gene environment interactions, because I think at least many of the susceptibility genes probably interact with environmental factors. Some of them may interact with the autoantigens, but many also with environmental factors. That's at least my personal bias. And then we go to that, how to study the environment and the interactions between genes and environment. That's quite difficult actually. And I think we should take much broader perspective here than before. And I like this exposome concept, which means basically everything to which we are exposed during our lifetime. And so it's like the counterpart of the genome and exposome. So I think we should go to that direction very much and look very widely these interactions. And I think there are probably risk factors in the environment and also protective factors. And Jill Norris already mentioned a couple of them, and I think that those are very good examples. And one of the, I think, opportunities here is also the subtypes and the heterogeneity. It's also a challenge, but it's an opportunity. And we already know that we have the different so-called endotypes, both in prospective studies and when we have looked at pancreas pathology, there are clear subtypes of the disease. And that's, again, a big opportunity for us to understand the interactions and gene interactions. So maybe this is my 5 cents.
That's good. That's great. Thank you very much. And how about Lu Ping?
Okay, well, thank you for invitation. So as echoing the comments from all the previous speakers, indeed the genetics and trigger was the environmental trigger play a really important role in type 1, no question about that. As Dr. Rich pointed out, that the genetic factor probably play up to 50% of the risk, and on top of that, probably the HLA genes have been shown for many years that play probably 50% or more of the risk, genetic risk. So this is pretty well established. And however, they actually even we understand that the several major HLA alleles and their importance and so forth. But there is actually, we stopped at the kind of a little bit short, just knowing those DP8.1, DP2.5, so some of them may high risk sort of the allele variants per se. The part of the lack of details, the understanding is that probably HLA gene is one of the most polymorphic genes in the genome. And of course, those genes are in a high LD with each other, and each gene carry a lot of rare variants, rare alleles. So making to study that a little bit difficult. Now, being trained in biostatistics and more mostly data mining, so one of the my interest is essentially really try to understand this one. Now, this become particularly interesting, important as we now have much better the next gen sequencing technology that actually really allow you to get a really high resolution of the allele, more importantly, to actually get the sequence directly. So we actually utilize those sequence data to be able to then try to examine not only just at a look at high in terms of the actual sort of the allele, but really going down to individual residuals. And I'm not going to do all that. We actually have several publications recently try to look at the class 2 genes with their association with the type 1. But let me actually focus on the one question being kind of touched upon by several speakers in the questions. Essentially, that as an example, the DRB1, especially DR4, is actually known to have this a little bit more complex sort of relationship. Some of the subtype is positively associated with the risk, and some of those actually negatively, actually protective, associated with the type 1. And no one knows actually why, except to give the name, this called yin-yang effect of DR4. And actually, we then actually use the approach we actually developed, this so-called hierarchical haplotype association analysis, and try to drill into it. It turns out actually it only requires several residuals and their motif. You can actually explain in certain context situation, it actually become the risk association and turn around, and the same sort of the residual has become protective. So that subtlety and details of genetic association really give some insight into how the class 2 gene basically recognize presenting those antigens, and in some cases not presenting it, and where the protection. So I guess we actually also look at the DQ, DQA1, DQB1, and look at the detail in terms of both the risk and resistant residuals have actually quite a bit of the findings and reported in the recent papers. And I would hope that as we move forward, as Dr. Rich sort of commented on the genetic important role, we look at it not only genome-wide, but really want to go back to HLA genes and try to drill a lot deeper into the HLA genes and to really understand how those the combination of the individual residuals actually really dictates the risk and resistance and mutual association with type 1. And more importantly, Dr. Marian and others have been sort of promoting that the type 1 is not single entity disease. It's really involving several convulsions and the hypoglycemia and the full-onset disease. This is potentially the disease stage of progression, for example, may involve different HLA genes as the several convulsion. So therefore, the approach which we're taking right now really is potentially also have implication of that. And then, of course, that if understanding those sort of the detailed association or the individual residuals and the motifs, and then we can actually begin to translate somewhat the genetic association of HLA to the clinical setting. Recently, we, as Dr. Vision and others, we actually have a paper specifically related to that as well. So there's quite a bit in interest to take some more novel new data science approach and to look at a more complex sequence data. Then we're hoping to get more insight into this complex but meaningful, interesting associations between HLA and type 1. Thank you.
Thank you. Thank you for that sort of summary about, you know, the sort of the intricacies of the HLA region. And I wonder if anyone would like to sort of weigh in at this point on what has already been said. I'd like to invite, you know, Doug Melton and Mark Atkinson to say a few words, and then we can sort of have a more, you know, interactive discussion. And then I will send out the second poll. Or Doug or Mark.
Maybe I'll begin with two questions. But first a comment. It's nice to hear from all these experts on the complexity of the disease and the appreciation that they're using really the most modern methods to try to analyze the genetics before I get to the environment. So that's, I think, very encouraging. As someone who doesn't work in that area, what I hear is that this is a damnably difficult problem, that there are multiple genes with small effects, that we don't know which genes and which cells they act lead to the disease. And the implication is there are many more than one way of getting type 1 diabetes. Like there are 64 different ways you could get the disease. And so if that's true, I would sort of just ask for people's predictions on whether or not anything has been learned from genetics that is actionable in terms of providing a therapy. So for example, we hear all the time about these HLA risk loci. My question is, since I haven't thought of how you would do anything with that, what do you do with that information if you want to develop a therapy? It's sort of hard to do this as a panel discussion. It's not quite like a dinner conversation. I might, Stephen's smiling, so I'm going to ask Stephen to answer the question.
Feel free to jump in.
Yeah, I mean, I was thinking, you know, Mark is probably one that could answer it better, but I think one of the key things for me is that, you know, HLA is obviously the gorilla in the room in terms of genetics. It accounts for about half of the genetic risk. There are people who have type 1 diabetes who have HLA, DQ0602 that has an odds ratio about 0.05 to 0.1, which means it's extraordinarily strong protection, yet they still develop disease. And so it's a question of what is it that causes something to overcome that strong protection. So that's why I think there's, we sometimes talk about twin studies as experiments of nature. In some ways, those who have 0602 and yet develop type 1 diabetes are experiments of nature, and that we can either hopefully use that information to define those perhaps genetic variants that help overcome that 0602 effect, as well as the environmental effects, and whether that's a specific virus that sort of triggers and then the genetics doesn't allow the appropriate response or not. I mean, those are the types of things I think we can learn from genetics. The other part is that I think it's important to realize that genetics is not destiny, especially when you have only about half the risk being genetic. And so it allows you to think about an overall genetic load, but what are the factors that either cannot modulate that genetic load? And that's where I think the environment becomes extraordinarily important, because the environmental factors can either increase or decrease the risk, just like genetic factors increase or decrease risk. So it's really the combination of the two, and knowing what the targets are, that things that Kyle has been working on, where you can perturb a system with cytokines or other types of factors, identify what those variants regulate, and knowing that it may not be the gene next door, but down the road, could actually, really inform what are the real true genetic factors and pathways that we may not have thought about.
Well, that's very helpful, but I'm still struggling. I mean, I do understand some aspects of this, that let's suppose we know that HLA is important and we know which cell it actually acts. If it's a dendritic cell or a beta cell, I don't care. What kind of therapies are you all imagining you would do with that information? That's what I don't understand. What is a therapeutic intervention?
So we are getting better at that. And let me give a quick 30-second preamble. I think we all have looked at the last 40 so years of studies on genetics environment and hoped that we were going to be reductionist, meaning we start out with a big list or start out with a big genome and then hone it down to very few candidates. But I think if we've learned anything, we're not in reductionist mode, we're in expansion mode. I mean, with Steve in the group's paper last week, now talking about 152 loci influencing diabetes susceptibility. And then now with 18 years of steady studies, we have more candidates than ever. So, the future, I think, as Steve sort of mentioned it, combining these into better phenotypes, and maybe, I don't know if AI has been oversold, but maybe with the hope is that with things like AI, if we can get the data sets available, that it'll help on here. Now, to your question about therapeutics and HLA, I think there's been a lot going on in terms of looking at responder, non-responder analyses in previous trials. Pretty much, we took all comers for clinical trials based on usually age, 7 to 12, 12 and up, 35. We didn't give as much attention to HLA. And I think now we have 3 to 4 trials that when they go back and look, either antigen specific or even things like anti-CD3, we're now seeing that people that say respond well to a GAD vaccine may be DR3s, or people that respond to a pro-insulin will be DR4. And even it came up at the FDA hearings about the DR4s driving the teplizumab might be better in those. And it could be part of their labeling when it comes out in July. Who knows? But I think we're scratching the surface now on this. It's a buzzword, personalized medicine approach. But I do think that HLA has the potential moving forward to help. But it's still early on.
That's very helpful. Thank you both. I mean, I still conclude that, well, I cheer you on because this is such a difficult problem. I mean, it seems so complicated. And seen from a distance, it seems to me that the kind of work Jill is doing doesn't get as much resources or attention. And if I was in charge, I would say, let's spend more money on environmental influences, given how difficult the epistatic analysis of the genetics has become. Monica, if you allow one more question.
Yes, absolutely.
I haven't had the privilege of meeting Marian before, and I hope he won't mind me putting him on the spot, but he said something that got my attention, which is he said, we need to have more sharing of data, that we have all of this data and no one's sharing it. That seems to me absolutely shameful and just completely inexcusable. So I would say to Marian, if you were in charge, why are people not sharing the data, and how would you force them to do so? It's really inexcusable that they don't do it. And I'm sorry for such an aggressive question on first meeting you, but I thought you'd thought about this.
Well, you are misquoting, Doug. Pleasure to meet you, but I have to say up front, you misquoted me so badly. I have to correct what you said. What I really said is that there are plenty of repositories, study-specific repositories, and some of them are hosted in the same building, an IDDK repository. And it applies both to data and samples. I didn't say people don't want to share. We do want to share, but it's not usually our top priority. When you come to your office, you have a list of stuff to do, and sharing data with Heikki, for example, is not my top priority. unless we are writing paper together. So I would like to give an example of PubMed. Remember how hard it was to get a reprint of a paper published in Diabetology or some other exotic European journal. You had to go to the library, find the volume, make a copy yourself, or send your graduate students to do it, and then read it. Now you have EndNote or other reference database, and just automatically, in most cases, upload in real time, even before the paper is published. So this is a great example of sharing published or publishable results. And there are now databases, Biomarker, I believe, where you can share preprints, data that have not been yet fully peer-reviewed and published, but still people want to show the world what they are finding. So I think Jdrf or Helmsley can do it piecemeal, but we really need to talk with the NIH and the government, where the big bugs are, to allow more sharing across the databases, and those discussions are already happening. We had a group headed by Carla Greenbaum over the past year, talking with Will Cefalu and others at the NIDDK, trying to get NIDDK, and I think we got a listening ear from their side to invest more in data sharing. And like with anything, there is a balance. Too much sharing, too much resources put in it may be wasteful, but it will be really important to prioritize, identify the data sets or repositories that are particularly helpful, and make the applications and the disbursement of samples and data easier on average investigator.
I see. Well, I'm sorry I misquoted you. I think I now understand. I understand you to say investigators want to share. What's missing are resources and incentives. And so if the NIH provided resources and incentives for this sharing, people will do it. Thank you.
Actually, we are facing a lot of disincentives. Zhao, Heikki, and others are part of Jdrf-sponsored tie-dye project. 5 studies, 2 from the US and 3 from Europe, I mentioned the names earlier, teamed up to share the data. We sent the data to Jdrf, our de-identified data sets. Jdrf facilitated collaboration with the IBM Watson, and we just published our first paper yesterday in Diabetes Care, but more are coming. There are 15 papers in the pipeline. So, on data harmonized, combined, cleaned from 5 different birth cohorts on 2 continents. So it is possible, but it's very hard. And what I meant by disincentive, European Union, in its wisdom, decided to really limit the flow of information and make it close to impossible. So Teddy has struggled for the past year to really make it possible for Heikki and Annette Ziegler and Åke Lermark to keep sending data from Europe to the US, to our DCC. I mean, this is reminiscent of all of the red tape that we've seen with RRBs and HIPAAs. It's just increasingly difficult to do any kind of human research, and especially share the data across the studies.
Let me just mention.
Could I also just jump in and Mark Atkinson was not on board earlier, and when I just mentioned that, well, my understanding was that also Dr. Rewers was saying that there needs to be a home for the data sharing, and and I stated at that point that like NPOD that was created at University of Florida by Mark Atkinson, maybe there needs to be a home for this sort of a hub for this sort of data sharing initiative. And yeah, I'd love-
Monica, can I jump in real quick?
Yeah,
Maybe
yeah.
Steve was going to say this as well, but
Yeah.
at least in the genetic side, there's there is a recently released repository and database for genetic data. Steve's involved, myself, our colleagues at the Broad Institute. It's called the T1d Knowledge Portal. So the goal is to collect genetic data, summary data, oftentimes things that can be easily shared in some cases. We're working on raw data as well. Other people who have data are welcome to work with us and share those data. So the hope is that that's a central hub for genetic data
There
for
it
the
is.
type 1 diabetes community.
So
So
you
it's,
guys have-
I'll send out the link as well. Yeah.
That's
Yeah, sorry,
fantastic.
Monica.
So you guys have established a home is kind of what you're saying.
Yeah,
So
it's
we're way
actually, I
ahead
mean,
of
it
you
was the
guys.
Broad Institute actually deserves most of the credit, but Steve and I and our groups were participating. But it's yeah, let me share the link right now. But so it gets released a couple months ago, and has data from our groups, and the hope is to collect at least on the genetic side and also genomic side, as much data as exists that's valuable for interpreting genetic signals and so forth. So if people have interest in sharing data, that would be phenomenal. So reach out to either of us.
Fantastic.
I was going to say that the NIDDK Center Repository actually has very comprehensive of all the data, and the susceptibles, and I have benefited greatly for accessing quite a few different valuable resources for some of the projects I'm taking on. So I think the data sharing is actually very good. I mean, in comparison with some other communities that I have experienced. So I think it's very good.
That's good to
Glad
hear.
to hear
I
that.
interrupted Dr. Rich earlier. Sorry, I didn't know if you wanted to weigh in.
Well, no, just again, Kyle is also bringing in all the epigenomic data as well, this transcriptomic data. So it's more than just genomic data. And I think the genomics community has always been good about data sharing. And this is just an example. And hopefully it's in one spot where people can get access to the portal easily. Hopefully, Doug, that will help you and your colleagues as well.
Dr. Norris, did you want to weigh in on how this kind of portal might be useful to your team and your perspective, your approach?
Yeah, I mean, of course, it would be extremely useful. I think there's an issue with regard to sort of curating it or making it accessible and understandable and really describing the variables. I mean, I would say it's difficult with genetic and genomic data, but it's probably more difficult for sort of exposure and environmental data because of all the other factors that measurement differences, et cetera. But I would like to agree with what Dr. Melton said, is I do think more resources should be spent on improving our ability to measure environmental exposures. I do think that we are lagging behind in some of that information. It's incredibly difficult because you're factoring in timing as well as instruments and et cetera. But that being said, it should be shared. If we're going to be putting that amount of money into refining exposure measurements, this is data that should be shared because not one single site can collect enough to really answer all the questions that we're asking.
So can I make one rapid comment to what Doug said? Doug, my opinion is there's different buckets here. One bucket is DKNet. So our millions of our tax dollars have been spent on DKNet. They had this vision for creating the NIDDK, one-stop shopping for efforts. And it's, if anybody on DK is on the call, I'm sorry. I mean, it's been pretty much a bust. But then there's the bucket of success stories. Like as mentioned by Steve and Kyle, genetics has long been into data sharing. Part of it's how you set up an organization. Thanks, Monica, for mentioning NPOD. We're a service organization. We're designed to share. The HERN around the HPAP thing, it's a service organization designed to share data, meaning it was set Pancreatlas, this Helmsley Trust initiative, where we're bringing all of the world's images on human pancreas together, designed to share.
Yes.
And then there's this third bucket that are the consortium buckets. And these are, I won't name them, but there are people are on there. But when you have multi-center consortium efforts that are designed to ask questions and then look at them that way, they're not necessarily or didn't have a priori the notion of upfront, or if they're going to share data, it'll be after it's been mined and papers published and anything. So I think it's hard to say there's no data out there. I think there is, but it kind of depends how the organization is set up. That's what
I agree with that. I think that I think there's quite a bit of siloing that remains, but there's a lot of effort to try to connect it, and with Kyle mentioning the T1d Knowledge Portal. That's an initiative that seems really ripe for sharing. And just to sort of a nod to the DKNet, we are trying to help them kind of bring them into engagement with the community. I think that's what they need, and actually connect them with other resources like the SSP and really with use cases of bioinformatics. And I'll talk about that at the very end of this, but I agree, they have a treasure trove of tools that are kind of in a closet, but we're hoping that they can be spread out and used with the world. I have another poll here. This is a great it's been really great, but so I wanted to ask one more question here. Oh, I want to share this. I guess we answered them all. And I want to share this. The second poll was, is it important that the scientific community merge global databases to create a more illustrative data set of genetic diversity in T1d? 95% said yes. So that kind of just, goes right on, is layered onto this discussion really. And then how can we create a better infrastructure to collect more information on T1d cases? And it looks as if people, the answers were, the selections were partner with hospitals, partner with T1d Exchange, partner with industry via strategic partnership, create a new entity like NPOD and other. And the top answers were partner with hospitals and for T1d Exchange. So I think that's kind of interesting in terms of collecting the raw data. And then I guess then following that, you create something like NPOD in terms of genetics, that is a repository, built to share, right? And that's what you were talking about earlier. So I think this is just, it's really been an excellent discussion. I'd love for a couple more people to weigh in about thoughts on how to best build these shared genetic portals to sort of have an end-to-end collection of the data in a clinical sense, and then partnering it with an open source, I guess, repository. Should you build it like an NPOD, or what are you envisioning?
Well, yeah, just to comment on the Type 1 Diabetes Knowledge Portal, a lot of the consortia have contributed data to that already from the genetic standpoint, including Teddy and TrialNet and Daisy, that have genotyping that has been utilized, or in TrialNet's case, not really utilized, but have allowed for analysis that can be then put into the knowledge portal. I think one of the real limitations that we have is the fact that in genetics, historically, most of the large studies have been done in individuals of European ancestry, and also in children with onset, whereas a large portion of those who have onset of type 1 diabetes are in adults, where we really don't know very much about them in terms of their genetic factors, if they're the same as in children, and also different ancestries. For example, we published a paper that was the largest collection of African ancestry type 1 cases and controls, and it still only had something like 1,500 cases and 3,000 controls. And so it's woefully short. And so you really need to have much more focused international, and by international, not just Europe, other continental collections that can be used for research. And that's going to be something that NIH can't do alone. It'll have to partner with other international organizations. And I think that would help bring us closer to understanding what the etiology and natural history of type 1 diabetes may be.
I mean, I guess I would offer that if we're talking about virus being a trigger for some people in some cases, there's a perfect opportunity here with the COVID situation. As some individuals get COVID and then go on to develop type 1 diabetes, wouldn't it be great to have access to those blood samples or those samples, you know, whatever samples that you can have access to, and then be able to look at those, and who might have those samples, who might, you know, be able to garner those samples. Anybody?
Well, maybe I could answer this. So I think one way is to use the biobanks, at least in Europe. The biobanks collect samples from hospitalized patients and also clinical virus laboratories. So they probably have quite nice repository of blood samples taken from patients who have an acute infection. The other opportunity is prospective studies like TEDdy, which collect samples during the epidemic, and then you can go to those and see how that influenced the risk of diabetes, for example. So I think this is a good example how these real life situations could be used to find the causes of the disease, and that has happened many times in the history, and they have been like coincidences. And those kind of things led to the discovery of, for example, the role of gluten in celiac disease. It wasn't just it was not scientific experiment. It was just coincidence.
Yeah, serendipitous. Would anyone else like to add anything more before I unveil our D challenge that we're calling it?
Well, I want to challenge Steve a little bit. Yes, we have 50, 60, maybe even 150 genetic variants associated with type 1. But I heard you saying on some occasions that many of those variants, we don't know the function. I mean, they are not causal. They just are in linkage disequilibrium with something that may not be a coding DNA, maybe just you know, a regulatory element, or you know, there's still a lot to be learned from a genetic point of view to figure out how it all works, including the MHC system. We think we know everything about the MHC, but we are learning new stuff, like the level of expression of those proteins coded by MHC genes seems to be even more important than the alleles. What are your thoughts here, Steve?
Well, yeah, I always go back to the fact that we've known about HLA since 1975. We still don't know about HLA. So it's something where I think the more science progresses, the more we'll know. The National Human Genome Research Institute is launching a major effort looking at variants to function across the lifespan in terms of development. I think that is probably going to give us a lot of information, given the fact that we're born with our genome and our exposures change over time, and yet the median age at onset of type 1 diabetes is like either at 10 years or at 25 years. So what is it that, you know, characterizes that progression of risk to disease at different time points in different people? And it may be the feature of development and the factors that go into the gene function at different times during development. So a lot of this, I think, as you said at the very beginning, Marian, this is a great time to be a scientist and to really be involved in research in this area, because the technologies are improving. The resources that we have to apply those technologies are becoming more available. And the intent on sharing information among the scientists it's also improving. So I think we'll find out much more, and it may confuse us more initially, but eventually we'll get to an answer, hopefully.
Yeah.
Could I make a quick comment about the data sharing, because that I think is very important.
Yes.
And I try to be very quick, but I think we discussed a lot about the raw data or original data and that sharing. But I think we should also consider changing the concept how we publish the findings in journals. So if we could engage the artificial intelligence so that we could, for example, have a format where each investigator, when they publish, could put the key findings in a way that the computer can read them, and then engage all this information, public publications, to a system which can really digest it. And I'm now thinking not only one discipline, but the whole scientific field, because I think that could actually invent completely new discoveries and completely new hypotheses and completely new openings. And it's a long way and far shot, but maybe some NIH or some maybe Google could jump in and create this kind of system. So because human brain can't digest all this information we are generating now.
Yes, I completely agree. And I would also offer that, biotech startup like Immune AI, based out of Tel Aviv in the Bay Area, are starting to do experiments like that, as well as Immune ML that are in the Netherlands. They are in Europe, they're trying to also attack this. And on our small scale, we are also trying to do it at the Sugar Science. So thank you again for all of our incredible panelists for joining us, and all of the audience for joining us for our third State of the Science, the role of genetic susceptibility and triggers in type 1 diabetes. Where do we go from here? Specifically, thank you for your time to our panelists, Stephen Rich, Marian Rewers, Doug Melton, Mark Atkinson, Jill Norris, Heikki Hyoty, Kyle Gaulton, and Luping Zhao. It was a pleasure to have you all speaking in a room together. And even though we can't be together, it had almost the flavor of a sort of a dinner party. So it was great. And at this time, I'd like to share our D challenge. So what is the D challenge? And here it is. Together with DKNet, the Sugar Science and Protocols, we are going to be presenting what we're calling the D challenge. And what is it? It's really a global focused scientific movement that sort of begins with the end in mind. We're leveraging our connections in type 1 diabetes research across the globe and inviting all of them to compete, to catalyze innovation and accelerate research towards practical cure for type 1 diabetes. So how do we imagine this might happen? The details are this. The D challenge is really a community research challenge. We're asking people, scientists, to form teams. And for the challenge, SPP, for those of you who don't know what SPP is the Signaling Pathways Project, run by Neil McKinnon down in at Baylor Medical School. And it's a multi-omics knowledge mine based upon public, manually curated transcriptomic and cystromic, meaning ChIP-seq, data sets involving genetic and small molecule manipulations of cellular receptors, enzymes, and transcription factors. Really, the goal of SPP is to create a resource where scientists can routinely generate research hypotheses or validate benchmarks relevant to cellular signaling pathways. And they've actually had some great success, where they collaborated with Index or NDX on coronavirus infection data sets in the recent past. So the SPP will really, for this challenge, the D challenge, the SPP will curate existing archive clinical T1d transcriptomic data sets to generate a T1d consensome and associated transcriptional regulatory networks, much as they just did for NDX and SPP. So if you're wondering what is a consensome, it's really a list of genes ranked according to a meta-analysis of their differential expression in publicly archived transcriptomic data sets involving perturbations of specific signaling pathways in a given biosample category. And consensome are intended as a guide to identifying those genes most consistently impacted by a given pathway in a given tissue context. So as part of the, the D challenge, these networks will be made available by the research community, or the group that comes accessible, sorry, by the research community. And the group or the team that comes up with the proposal that best exploits the networks to generate novel insights into aberrant signaling in T1D will win the prizes. We have $20,000 in prizes, and that money will be stipulated to be used for the teams to further research. I guess we also want to bring in the facts that DKNet and SPP will provide a data-driven roadmap, really, that you can use to design a working hypothesis to further research in your realm of T1D. And we're challenging the team, these teams to really use this sort of data-driven environment to formulate a new hypothesis and hopefully take a good advantage of this new hypothesis center that's been designed by DKNet. We will be partnering again with SPP as well as NDEX or NDEX, and with sort of the support system of the Sugar Science and DKNet. We're hoping that this challenge will be, widely received, and people from across the globe will sign up. So hope to see people signing up. We open registration tomorrow, and it will run through November 5th. So you have quite a bit of time to really get into the data and formulate your hypothesis. So it's very exciting times for us, and we hope you're excited about it as well, and that you'll register and form your team. Thanks again.